This is Dr. Henry Nasrallah, Professor of Psychiatry, Neurology, and Neuroscience at the University of Cincinnati College of Medicine. I will be giving a presentation in two parts. The first part is a brief overview of the human gut microbiota, both structure and functions. Learning objectives: first, to enhance our understanding of the trillions of gut microbiota and the factors that impact them. Second, to identify gut-related microbiota pathologies that affect the brain and trigger psychopathology, especially psychosis. And finally, to discuss possible therapeutic interventions into the microbiota and their microbiome for patients with schizophrenia and related psychosis. So why does the gut matter? Well, since the days of Hippocrates, the father of medicine, before about 460 BC, he said that all diseases begin in the gut, and his classification was black bile, yellow bile, phlegm, and blood. And of course, he had no idea that the microbiota existed, but he described what he saw clinically. Following that, Galen, after the birth of Christ, also pointed out something similar. So the human body is now known to host trillions of microbiota, and those microbiota are practically more than the number of cells in the entire human body, about 39 trillion bacteria and other microbes, compared to what 30 trillion in the human body. And as you can see, the microbiota are actually comprised of a variety of germs, bacteria, fungi, viruses, protozoa, and archaea. So all of those exist in the microbiota within the human gut in varying degrees. People talk about trillion all the time these days, especially with regard to budgets and so on. But if you count one number per second, it will take you 11 days to count to a million, 31 years to count to a billion, and 31,000 years to count to a trillion. If only people knew how big trillion is, they probably appreciate how many microbiota we have in our body. So the microbiota, or microorganisms, if you want to call them that, which share a lot of genetic material called the microbiome, are divided into 5 kingdoms: the bacteriome, the virome, the mycobiome, the archaeome, and the parasitome. So this is the way we call them to distinguish them from each other. Again, as I said, they exist in various amount and concentrations. And they interact among themselves and also interact with the human host. And they certainly impact physical and mental health. There are about 1,000 species of diverse bacteria, and they contain 4 million bacterial genes, and those genes are called the microbiome. So the microbiome is really the genetic material of the microbiota. Compare that to 22,000 genes in the human genome. We only have 22,000 genes that make protein coding genes. So you can see it's about 20,000 times more genes than the humans themselves. And it's an incredible density, actually, of bacteria in each cubic millimeter of gut. So here are some questions for you to ponder. What is the largest endocrine organ in the human body? And what is the largest immune organ in the human body? Well, the answer is the microbiome. It exists within the 30-foot-long GI tract, which houses the microbiome, which is the DNA of the microbiota, and also the enteric brain. It's called the smaller brain or the other brain, and that secondary brain also is housed within the GI of humans, and it's side by side contiguous with the microbiota, and they both interact. So here are the various forces that can impact the microbiome, ranging from diet, antibiotics, stress, toxins, environmental exposures, various maternal factors, including mode of birth, breastfeeding, and maternal flora, host genes, age, sex, gender, pets, exercise, sleep, food sensitivities, pro and prebiotics, and then the psychiatric disorders themselves, as well as physical disorders. All of them can impact the microbiome. Here are some of the food effects and the microbiota. There's a lot of emerging evidence that the food we eat can actually modify the gut microbiota via epigenetic mechanisms. These include methylation, which expresses genes or overexpresses them sometimes, and histones, which can suppress genes. And when that happens, it has quite a few consequences for humans, actually. And probiotics, which are actual germs, good germs, can exert epigenetic effects by influencing cytokines, which are inflammatory biomarkers, producing short-chain fatty acids and vitamin synthesis, and producing various neurotransmitters. So there's a lot of effect on probiotics on the microbiota. How humans get the microbiome? Fetuses do not have any germs while they are in their mom's uterus, but at the time of birth, when they traverse the mother's vagina and basically during normal birth, they acquire the vaginal microbiota from the mom, and it becomes the gut microbiota for the baby when they're born. So basically, all babies acquire the microbiome from their mom's vagina, but also after birth, when they start breastfeeding, they can get additional microbiota from the nipples of the mom, because there are microbiota on the skin. We're talking about gut microbiota, but there are also quite a bit of microbiota on the skin of humans. And so microbial colonization can occur during breastfeeding, touching, skin-to-skin touching, or from pets, as I said earlier. So what are the impediments to acquiring a healthy microbiome after birth? Well, C-section is a problem, because when babies do not go through their mother's vagina to basically be born, and they're removed directly by C-section, they end up acquiring the germs in the hospital air, which has a lot of bad germs, as you know. So it is recommended these days that the obstetrician take fluid from the mother's vagina and basically wipe the baby's skin with it in order to help the baby acquire a healthier microbiota than the air of the hospital. Bottle feeding also deprives the baby from acquiring additional microbiota from the mom's nipples, and treatment with antibiotics can kill billions of microbes in the gut, and that includes killing both the good and the bad ones, and disrupting the healthy diversity. Indoor living, excessive sanitation, and food preservatives all can have also some negative impact on the microbiota. So microbiota and the brain communicate regularly, and we call them the microbiota-gut-brain axis. They communicate in different ways. First, with systemic immune communication, which includes cytokines and bacterial metabolites. Also with neural communication, the vagus nerve is the highway that connects the big brain and the small brain in the gut, which of course interacts with the microbiota all the time. And the sympathetic nervous system has quite a bit of impact. And then systemic endocrine communications, including the hypothalamic pituitary adrenal axis, HPA, neurotransmitters, and the bacterial metabolites. So what is microbiota dysbiosis? Dysbiosis is a bad thing to happen. It means the loss of a healthy diversity of the microbiota, leading to an unhealthy balance. And that may lead to a variety of psychiatric problems, including mood, anxiety, cognitive, and psychotic disorders. So we want to try and avoid dysbiosis if possible. The causes of dysbiosis include, as I mentioned earlier, antibiotics, unhealthy diet, especially high-fat diet, which is inflammatory, and stress. All of that can result in dysbiosis, and the consequences include a rise in inflammation, altered behavior and cognition, and increase in gut permeability, and that is called a leaky gut. That's a very pathological situation where basically the microbes leak from the membrane into the body and can cause a lot of inflammation. That basically ends the overview of the microbiota and the microbiome. Thank you for your attention. I hope you can log into part 2 and basically hear about how the microbiota can basically cause or contribute to psychiatric disorders, especially psychosis.